So here we are on Lower St. Regis Lake. The topic of this lab that we're going to do here is about kinds of living things on Earth that are not animals and not plants, but micro creatures. Some of them actually are animals. They're about the size of a salt grain. We'll see some of them. But most of them are classified in the other kingdoms of life. Protista, a little bit in fungi, and also in bacteria. So if you look at a place like this, normally when we look at a lake, we'll say, well, what a nice place to go for a swim on a hot day. What a nice place to put a boat. Or what a nice place to reflect the sky and the scenery or something like that. But the story of the lake is under that surface, and there's a lot of life here. You can kind of tell that once you get used to this topic just by looking at the water itself in the shallows. And if we sort of come close to the edge and you look down in there, you might see there's kind of a brownish color to the water. Well, part of that is the sand down there. Part of it's a little bit of organic matter coming from the local soils. But a lot of it is because there is life in the water that has a kind of a brownish or a greenish color to it. Otherwise, if it was pure water, it would look like a swimming pool. So this alone tells you this lake is alive. But you need special equipment to see this. So we're gonna take a look through microscopes later, but first we're gonna collect real wildlife out in the wild that's living in this lake. And in order to do it, we're not gonna use fishing rods or spears or things like that. We're gonna use a plankton net. All this is a material with very, very fine pores in it. And as we pull this net through the water will go in here, come out through the mesh on the sides, but living things that are in the water will be trapped inside, and they'll collect down the funnel, and then we'll put them in a jar down there. So the way we do this is sort of treat the plankton net as if it were a whale in the ocean filter feeding for krill or something like that. So I'm going to fling this out into the lake. It's a little bit breezy, so I'll throw it with the wind. I'll stand on the rope so we don't just fling the whole net out into the lake and lose it. It's actually expensive. You sort of fling it like this, let it go out, and then you pull it in kind of nice and easy. Not too fast, because then it'll just skip over the surface. You don't want it to float too low, because it'll scoop up dirt. And here you can see the water running out the sides like that. Now, there will be some living things in there, but I'm going to do this several times. The more tosses you do, the more concentrated the stuff's going to be. So, do a few of these. We often have students volunteer to come out before class and do this. We always do it in the same place, here on the point. We ask you to come out in teams of 2, so one person can hold one end of the rope and the other one can fling the net. And then bring some samples back to the lab where the rest of us can use the microscopes and see what we caught. One of the great things about doing this is that we not only do it once to show you real wild microbes, but we also do it through the whole semester. And what we find is the things that are alive in the lake will actually change through the seasons, just like plants do on land. You go to a meadow at different times of year, you'll see different things blossoming at different times of the season. Same thing happens here. But if you see little flecks down below, those are animals. The technical name for that is zooplankton, which means animal plankton. Animals that are so small, they drift with the currents instead of being able to resist them very much. So a fish will just dart wherever they want to go. These are so small, the smallest currents just push them around. Mingled in with them that are too small to see will be tiny algae and tiny bacteria. They're called phytoplankton, which basically means plant-like plankton, which is so tiny that it also drifts in the water instead of swimming or something like that. I'll do a couple of more tosses and then we'll see what we got. You know, I'm going to squeeze out as much water as I can. That will concentrate whatever we catch into a smaller amount of water. And then I'll put it in a little jar and we'll see if we can hold it up to the light. You'll get a sense of what we got. Alright. Now, if you're the one who's collecting this, by the way, it can get a little tricky. Here's a little catch on the bottom of the tube. You open this up and the water will come rushing out. Sometimes it's a little tricky. It doesn't want to let go. So we'll see what happens. I'm going to put it into this bigger jar, and if it looks good, I'll put it in a glass jar so you can see it better. So you just sort of put it in there, and then this is why you want to have two people doing it. I'm going to hold it down here with my feet, and I'm going to unsnap this, and there goes the water into the jar. And if you look down inside, you should be able to see lots of tiny little specks. There's actually an insect trapped in there too. I'm going to pour some of this into a little glass jar. You should see little specks that aren't just drifting, they're bouncing around in there. These are tiny little animals, the zooplankton, that are related to shrimp, actually, even though it's fresh water. Like you said, they're about the size of a grain of salt, a grain of pepper, but they're bouncing around in there. They're trapped and they know it. These have never been confined in their lives before and they don't know what to do. So they're bouncing around in here. When they're not freaking out like that, they would be out there eating algae, eating the floating bacteria, sometimes the fungi, sometimes each other. There are many different kinds in there that you can see under the microscope. So I'm going to set these loose, so we'll have saved hundreds of little lives by doing that. But we are going to take one sample back to the lab so everybody can take a look under a microscope. Just think of all the little life forms that you're swimming among anytime you swim in a lake like this. So if you're the one collecting this, you want to make sure the cap is on the jar. Use the plastic one because it's less likely to break on the way back to class. You roll up the string so it's nice and neat. And we also want you to take the temperature of the water. And the reason for that is it's also part of a long-term study that we've been doing with Biology 101 labs for the last 30 years. We've had bio labs coming out here to do what they do every fall since like 1990, something like that. We've got the data that go back all those years, and we're monitoring whether the plankton changes at all from year to year, and also is the temperature changing, and in fact we're finding that it is. So we're going to have a little thermometer for you as well. I'm going to ask you to pull it apart and lay it down somewhere where it's not going to wash away, but we'll get a good temperature. So you can either leave it lying there, or better yet is hold it if it's not too splashy and stuff like that. You leave it in there for 30 seconds, something like that. The water's pretty warm right now. We're going to measure this in degrees centigrade. So here I'm seeing 27, it's 28 degrees centigrade. So that's a little hotter than room temperature. So this would be the assignment before the lab actually starts. Come out, take the sample with the plankton, take the temperature of the lake on the surface, write it down so you don't forget it. Come on back into the lab and we're going to see what's alive in there. So we're on the other side of the point now, having taken a plankton tow and measured the temperature of the water. Here on this side, just want to review again what this lab is about and what we hope you get out of it. There are two parts to it really. One is to expose you to the diversity of life on Earth, especially on the small size scales. So we'll be looking at what microscopic life forms can you find, in addition to things like this robber fly or the little whirligig beetles on the surface of the water. There are things you need a microscope to see. So we're going to be looking at plankton, and also contributing to a long-term monitoring study that's been going on here for several decades, measuring environmental change as the world changes from invasive species and climate change. So just to review what plankton is again, basically it's defined as something alive in the water that's too small to really swim very fast. And so the currents can kind of take it wherever they will. It can be sort of like an animal, it can be sort of like a plant, but it's still called plankton. If it is a small animal or resembles an animal, you call it zooplankton. Zoo means animal. So that would be little water fleas, like we were saying, about the size of a grain of salt. We'll be seeing those under the microscope. They're related to shrimp, but also protozoans and things like that would count. If it's sort of like a plant, meaning that it traps sunlight and does photosynthesis, that would be algae or bacteria mostly. So they're called phytoplankton, and we'll see them also. So what's worth monitoring this from year to year and keeping all the data? There are a lot of things you can use this information for. For one thing, as we'll see by taking samples through the whole semester, the lake itself changes with the seasons. So there will actually be dramatic enough changes in the plankton that the color of the lake will change a little bit, and we'll see that as we go. But it can also change from year to year as the environment changes. So what we're learning here is not just theoretical about the diversity of life, not just theoretical about environmental change, but it's also something you might make a profession out of someday. People use these kinds of living things to monitor environmental change for consulting firms or anybody responsible for managing a lake and things like that. So it's a great opportunity to do that. So one of the things you can monitor is the quality of the water. How clear is it? People tend to like clear lakes more than murky lakes. Is there some kind of plankton growing in there that's bad for human health? That can change as well. Is the lake polluted with something that makes certain kinds of organisms grow in it? So by learning the kinds of plankton, you can monitor water quality in a lake over time. We had a history of pollution on this lake years ago. The college's septic system used to leak into the lake, and people were also doing that kind of thing before that. So we want to see, is the lake recovering now that we've stopped polluting it like that? Is it getting better water quality over time? Another thing we can watch for is invasive species. You might have heard of, certain kinds of animals or plants moving in, water milfoil, let's say, or animals like maybe an exotic kind of a mosquito that brings Zika virus or something. But you can also have invasive microbes in lakes too. One of the things we might want to be watching for through the years is the arrival of new kinds of zooplankton that can change the food web. So just by monitoring what's here from year to year, we're keeping an eye on that so that if invasives like that do show up, we'll know if they're changing the ecology of the lake. Another thing is climate change. We're seeing it for sure here in the Adirondacks. The weather stations show that it's getting warmer, but also people who keep track of the ice on the lakes are seeing that over the years, the ice is staying on the lakes less and less time in the winter as the winters get warmer. The lakes are freezing later, melting earlier in the spring, and that can affect the ecology of the lake. Sure enough, if we look at the temperatures we were getting in this lake when we first started this study decades ago, the temperatures on average are actually warmer than they were in the past. So we're actually documenting the effects of global warming right here on our own lake. And by monitoring the plankton too, we'll see if that's changing the ecology of the lake at all. And finally, I'm just looking here at what you have on your lab handout. Again, it's important to remember that we're not just looking at pretty interesting, fun things under the microscope, but we're classifying them as well, putting them in the right kingdoms of life and understanding what they have in common and what they have different. So we'll be seeing things like algae and cyanobacteria, which can look similar but actually are very different ecologically, and they belong to entirely different kingdoms of life. So there's a lot to cover in here, but it's always fun to do it in a beautiful place like this. So here's a microscope of the kind that you'll be using to look at the plankton. This is a pretty powerful one. It's also a kind of a delicate and expensive piece of equipment. So when you're carrying this to your bench, make sure you have your hand underneath and have a good firm grip on it. And just place it gently, so it's not right on its own electrical cord. Find an outlet and plug it in. You'll normally be sitting. I'll stand just to demonstrate it here. So the basic principle is there are two sets of magnifying lenses for you. There's a set here called the ocular lenses or eyepiece lenses. They magnify whatever you're looking at by 10 times. When you look through that, you can use both eyes to do it as if it were binoculars or something. When you look through that, that image is going to be funneled up through another lens down here. And this is called an objective lens. And it's much more powerful than these oculars. So the sample's going to be here on this flat part called a stage. There's going to be a lamp underneath that you turn on back here with a little clicker, and the light comes on here. Then there's a red knob that you can adjust the brightness for. Over here, you just sort of roll the knob. You don't want it totally bright like that and leave it because it'll burn the bulb out, and it's also just really too much. So you want it somewhere intermediate brightness. That light is going to shine up through here, through your sample, which is going to be here, up through the objective lens, and then into your eyes from the ocular lenses. So in other words, you got these tiny things here, they get magnified by this, and then they get magnified again here. The way you tell how much it's magnified is you multiply those two together. This is 10 times. Depending on which lens you use, it can magnify it 10 times. In here it says 10x on this one with the yellow band on it. 10 times 10 is 100. Anytime you look at something through this, the way it's set up now, it's 100 times bigger than normal. But we've got two other lenses you could use too if you wanted. One of them here, you can swing it down in. It's got a blue band, and on here it says 40x. That would be 40 times 10 is 400 times. And then there's like a real monster lens here. That's 100x times that. It's 1,000 times magnification. This one, you need some oil to put on it and things. We're not going to use that. You want to use, for starters, this yellow one, the 100x total, or the blue one, 400. I'd recommend starting out with this yellow one. It's easier to find things. Now, the more magnification you use, the more light you need. So if you do this and you look through here, it might be too bright to actually see much of anything. So you can adjust the brightness to match your eyes. So you can either fiddle with this red knob, or even better, you'll feel under here or look carefully, there's a little lever that you can push side to side under here. It's called a diaphragm. And you should mess with that because I'm going to push it side to side. And if you notice, if you look in here, you see it gets really bright. That's because the hole is totally open. And as I move the lever, I can shut it down so there's even less. When it's kind of shut down, it's actually way easier to see things. You see more contrast. So don't forget to mess with the brightness and this lever here that opens and closes the diaphragm. If your lighting is good and the lenses are clean, then you should be set to go. So here's some other things. You want to use a glass slide to put your sample on. So you'll have something like this. let's say here's a sample from the Plankton Net. You take a little eyedropper, you squeeze out the air and just pull up some stuff. The plankton tends to sink to the bottom, so if you do this, squeeze out the air, push it kind of near the bottom, and you'll get a nice juicy pile of plankton that way. Let me repeat that, by the way. It's common for people to have this and you put it in there and then you squeeze the bulb, then you're just blowing bubbles. You're blowing the sample away. So squeeze the air out first, pull up your sample, and then just put one drop, maybe two, onto the slide, and then you pin it all down with a tiny little super thin flake of glass called a cover slip. And you just plop it right on there. It flattens the drop out so it's nice and uniform, and all of your little plankton organisms are now swimming in a little thin layer of water under that. So you take that, put it on your stage, and then you have this really fancy mechanism for moving it around to find things. So you take this here, and it grabs onto your slide. So here it is again. There's a little lever, moves that little mechanism. You can push the slide in and it holds it in place. Then you have these two knobs. One of them moves the stage this way, and the lower one moves it that way, side to side. So the great thing is your samples are in that little square. You want them to be right over the light source. So I'm going to center it there, put it right in the center. And as you're looking through here, you can use those two knobs to move it just the tiniest amounts, and you can follow little protozoans as they swim or hunt through the whole slide systematically to see whatever you need to see. So the last thing is you want to focus it while you're looking in there. So you make sure it's centered. In fact, you could even swing that objective lens away to make sure it's kind of in the center like that, and then swing it into place. It'll click, so you know it's there. Then you want to use this big knob to focus it. So you start by turning it, and if you notice, as I turn this, the stage goes down and then it comes up. That will focus what you're looking at. So usually you start with it as high up as it'll go. So I'm turning it here like that. And then as you look through, you slowly turn that big knob. The stage will slowly lower down. And if you do it gently enough, all of a sudden, a bunch of things will pop into focus. And if you go past it, well, you just twiddle the knob back and forth and it'll go in and out of focus till finally you're kind of there. Then you use this smaller knob in the middle to finally focus it like that. And if you've got the lighting right, it's not too bright. And you got it positioned right on here, so it's right over the light source. And you move these to put it right where you want to see it. You focus this, you should pretty much be ready to go. Then you're going to want to hunt around for zooplankton and diatom algae and cyanobacteria and all kinds of really cool things in here that you may have never seen before, but are really common in lakes like ours. One of the great things about this is in most biology courses, you'll learn how to use a microscope. You'll learn how to look at microbial life like this. But the great thing about the way we do it here is these are real wild creatures you found in a real lake right where you live. So instead of just ordering amoebas in a jar, we're going to go out and see what really lives out in the wild in our lake. Keep the information, add it to what students have been doing for decades now, and contribute to a long-term monitoring study of the health of the lake. Sometimes people have trouble focusing. Well, one of the reasons might be maybe the ocular lenses are not matched to your eyes. So they actually move like this side to side to match the spacing of your eyes. So if you look in and just kind of get them to line up, you should only see one big circle of light, not two. If they match your eyes, your brain will just make it into one circle. So just mess with the spacing of this. Also, sometimes people's eyes are different magnifications. Mine certainly are. So as you look through there, you could adjust a little bit of focus with this as well for that eye or that eye, if that helps. Ask someone to help you focus just to get started, but once it's in place, you should be fine. Another thing is, if you want to see something up close more than you already have, you can change which lens you're looking through. So this objective lens can just be swung out of the way, and that higher magnification one will be there. And if things are done right, it's already in focus for you. Just maybe adjust a little bit with that fine focus while you're doing it. The other thing is, maybe it's blurry because the eyepieces, the ocular lenses, are dirty. So you always want to make sure they're clean. There's a special kind of paper called lens paper. It's not Kleenex, it's special microscope lens paper. You just take a little corner of it and use it to just puff onto here to dampen it, and then just rub that on there and clean it, and that should help. You could even do that to the objective lens if you wanted to, just to make sure that's nice and clean. But just remember, only use lens paper on these. If you use regular papers, they can actually scratch this expensive glass, and you don't want to do that. So last thing is, well, you want to show your instructor or your neighbor some cool thing you found under here. What's common to do is to say, hey, come check this out. And then you kind of are standing aside, and this person comes up to see it, and you want to turn this to help them see it. Well, these have pads under here to grip the table. So if you try to slide it, it's going to go boom, boom, boom. I'll do it a little bit now, like that. If you do that, you just jiggle it so hard that whatever you're looking at is now out of view. So you don't move the microscope once it's there. People move, you get out of the way, whoever it is you're showing it to comes in and takes a look and does it that way. When you're done, you turn off the light, just click it off, you unplug, you rewrap the cable. You, if you're polite, will also clean your eyepieces with a piece of lens paper for the next person who comes. You make sure this is off the stage and put away. You may throw it out or you may wash them. You pick that up and use 2 hands and take it back to the cabinet nice and gently. And there you go.